Hey, what's up, guys? This is Audie from ShastryNation, and today we're going to be talking about enzymes. Okay, so now that you're back from the intro, let's go ahead and get started. Okay, so what are enzymes? Enzymes are basically proteins that speed up reactions. So they speed up or catalyze reactions. Yeah, so they speed up or catalyze reactions, and they are used by our cells to, just speed up reactions, lower the amount of energy needed to do things. So they also lower the energy, I forgot to mention. Lower the activation energy. So these are basically, activation energy is the amount of energy needed to start a reaction. To start a reaction. So yeah, this is the amount of energy needed to restart a reaction. And so how it does this is by binding to the enzyme. So the substrate will bind to the enzyme. Substrate will bind to the enzyme. And the enzyme will use this thing. The enzyme can also use this thing called the induced fit model. So a substrate is basically a reactant in the reaction. Go ahead and write that. Reactant. Or the thing that's being catalyzed basically and starting to react. So basically, if I drew a circle with a pie cut out of it, and then this was the substrate, this would go into this, and this would be the enzyme. And so this would be the substrate. So yeah. So what is the induced fit model? Let's go ahead and talk about that now. The induced fit model is basically when the structure of the substrate is changed by the enzyme. Of the substrate is changed by the enzyme. And so by changing the bonds and the structure of the substrate, the enzyme can then make it more likely for the molecule involved to start a reaction with the other reactants that it needs. And so an example would be an example of an enzyme, not of this, but an example of an enzyme. And now keep in mind that there are hundreds of thousands and millions of enzymes in the human body right now as we speak that are working to do the day-to-day tasks that we do. So an example would be this molecule called Rubisco. This Rubisco is an enzyme. So Rubisco is an enzyme in the Calvin cycle and is used to take apart, and it's used in carbon fixation and to take apart the parts carbon dioxide to use elsewhere in photosynthesis. So basically what it does is, if you want to know exactly what it does, you can go and watch my video on photosynthesis. I'll have it in the description, and I'll have it here around this photosynthesis if possible. If not, it's going to be in the description. If I don't remember to put it there, it's going to be in the it's going to be in my videos. It's like the first video I've made. So yeah, now that we know what enzymes do, let's see how they're affected by the environment. So let's see how things affect an enzyme. Okay, so how things affect an enzyme. So let's talk about temperature, and then pH, because these basically show the same thing. So an enzyme needs to be in a perfect environment for it to work, because it's made of proteins. And if the environment isn't maintained to a certain degree of perfect, to a certain degree, the enzyme will become denatured. So denatured. And so this just means the enzyme won't work. It'll lose its shape and it just won't work. Won't work. So by losing its shape, it won't work because the enzyme can't like take in the substrates and have it react, basically. Okay, so let's go ahead and talk about how temperature and pH affect this. So temperature and pH both affect the enzyme in the same way, and so the enzyme will have a perfect temperature and a perfect pH level that it needs in the environment to react at its optimal conditions, like to react in its fastest way and to catalyze the most reactions and stuff. So the graph of this can be seen like this. So you do this and then it goes like that. So basically this would be the perfect temperature for it to react in. Actually, that's a pretty bad graph, actually. So yeah, so it would be like this, like way steeper, and then it would go like that, basically. Right. So this would be the optimal temperature / pH. Optimal. And this would be the temperature increasing. This would be the rate of reaction increasing. So as you can tell, the temperature and the pH have to be exactly what the enzyme needs. I'm not going to say that the enzyme will not work here. It'll just work really slowly until it hits no reaction rate at all. Okay, so then let's talk about the amount of enzyme. Amount of the enzyme. Or and we can also talk about I can just say / substrate, because these also, like the pH and temperature, change the reaction rate in the same way. So let's see. Now we have in this graph. Let me just show you the graph because it's easier to explain. So in this graph, it starts out like this. It goes high and then it goes like that. It levels out basically. So the amount of enzyme, while it will make the reaction go faster because there's a lot of things working, but then what happens is that once the enzyme, once there's too many enzymes, the amount of product just gets in the way, and the enzymes start reacting with the wrong things, and they just lose control because there's so many enzymes in that same place, and they just can't keep track of what's being reacted and stuff like that. So it makes sense. And then substrate, if there's a lot of substrate, the enzymes will react faster at first and make more product. But then if there's too much substrate, then the enzymes just can't get to all of the substrate at a specific time, and they need to just slow down. All right. So those are the things that affect the enzyme. Now let's talk about things that like really affect the enzyme, like chemicals and stuff like that. So chemical. So there are a couple of types of chemicals that affect the rate of reaction, and so some of these are called inhibitors. So inhibitors, what they do is they destroy some of the functional groups on the enzyme, and then it renders the enzyme useless. So destroys functional group. And destroys, and the enzyme is useless. The enzyme is useless. Right. So then yeah, the enzyme is useless. Okay, so then the other thing is reversible. Reversible inhibitors, and this does the same thing, but it doesn't destroy the enzyme per se. So it doesn't destroy the enzyme. And then we have competitive molecules. Competitive molecules, and these are just molecules that are in the same shape and can bond as a substrate to the enzyme, and then compete for a spot. With the actual substrate or the molecule that needs to be reacted with. It'll compete for a spot on that enzyme, and it will obviously slow down the amount of reaction, the rate of reaction, because there's just not space. A lot of the enzymes are taking up competitive molecules that don't do anything. So these just compete with actual substrate for a spot on the enzyme. So yeah, and then there are things called non-competitive. So non-competitive. And so these also bond to the enzyme. But they bond in a different space, in different places, and the enzyme will lose its shape. So bonds to different places, and enzyme loses its shape. So yeah, then it loses its shape. So another thing is this thing called allosteric regulation. So it's a type of regulation where this molecule will turn the enzyme on or off. When it needs to. So this way the enzyme's just not working when it's not needed, basically. One way of doing this is called feedback inhibition. It's called feedback inhibition, and basically what happens here is that the product of the reaction, of the catalyzed reaction, bonds back to the enzyme and then stops the reaction from occurring. So product stops reaction. So the product stops the reaction, and then what happens is we have so an example of this, we can see this in the production of ATP. So production. So ATP will go back and bind to a very essential enzyme in the process in cellular respiration. and it'll stop the reaction from occurring. So yeah, that's about it for enzymes. So basically, enzymes just speed up reactions, and they bond to the substrate, and that helps them speed up the reaction and stuff. So yeah, it's not that difficult, I guess. It's pretty easy. I guess that's all I needed to say about enzymes. Hope that helped. And be sure to subscribe, because I make videos like this weekly. And yeah, I guess I'll see you guys on the next one.